Okay, we'll go ahead and get started. So it's my pleasure to welcome you all to Plant Phenomics Fridays. There's my Chicago accent coming in with phenomics. Sorry there. Plant Phenomics Fridays, from satellites to seeds. It's our spring seminar series. I'm Dr. Nick Searing. I'm from Statistics Department, just filling in for Dean Carolyn, who can't be here today. We have two speakers joining us today. It's Dior Kelley and Alex Bucksch. We'll aim for about 20 minutes or so talk, maybe a couple of questions, and then after both talks, we'll have a short discussion session. Before we introduce the speakers, I just wanted to recognize the supporters of the Plant Phenomics Fridays series. HIPS, HIPS, the High Intensity Phenotyping Sites, Multi-Scale Multimodal Sensing and Sense-Making Cyber Ecosystem for Genomes to Fields. That's funded by an award from USDA NIFA. And also IRA, the AI Institute for Resilient Agriculture, funded by USDA NIFA and a grant from the NSF. So now I'll go ahead and introduce Dior. Dior Kelley is an assistant professor here at Iowa State University. Dior got her BS in chemistry from UC Santa Cruz and her PhD in plant biology from UC Davis. She did her postdoc work with Mark Essel at UC San Diego before joining the Department of Genetics, Development and Cell Biology here at Iowa State University. Her laboratory studies the hormonal control of root growth and development in two model systems, Arabidopsis and maize, using systems genetics approaches. Dior.
Thank you. Okay, great. So thank you for the introduction. Thank you for the ability to come and talk today. I really appreciate it. I'm going to talk about all unpublished work today. And if my volume is too loud or too low, somebody's going to have to let me know. And this is really what I also like to call sort of a pandemic project, because it really was started during the pandemic and has been work that has been largely spearheaded by two undergraduate researchers in my group. So, most projects that people are familiar with these days are highly collaborative. And I wanted to start by saying thanks to everybody who has been, really fundamental to making this project happen. So really this started a couple years ago when Justin and Nick and I went and had a beer at Alluvial during the kind of start of the pandemic. And we were sort of brainstorming ideas and what we might do if we were going to be allowed to actually work in the field that summer. And one of them was to do a really big genome-wide association study and look at different hormone responses. And of course, we're going to start with auxin because that's the hormone that I kind of tend to love the best. But I will have to say that if it wasn't for Nick being such a positive kind of like, yes, let's go for it and do it, it's probably something that I wouldn't have taken on, completely independently on my own. And then Brian Dilks, who's at Purdue, has been a longtime collaborator, who's also been really great in this project. And then, the meat of the work has really been done by two undergraduates, Melissa and Becca, and then also a postdoc in Brian's group, Raj. And so that's what I'm going to present today. We're very much indebted to Miriam, who has been kind of the organizational mastermind behind the wheels for especially bulking up the Wisconsin Diversity Panel 2 years ago, which was a ton of work by many people. And then also Sarah and Ludvin, who were two undergrads in my group, who were really important for helping with the phenotyping, because that was kind of a brute force. Okay, so in my group, we study this hormone auxin. We also look at other hormones, but this is our favorite. Indole 3-acetic acid, naturally occurring form of auxin, produced by plants and bacteria, really important for regulating pretty much every aspect of plant growth and development that you want to think about, or maybe not think about. Synthetic auxins are also of interest to us because they're used widely in agriculture and also in horticulture for propagation of things like fruit trees, anything that's clonally propagated. So two examples of synthetic auxins are 2,4-D and dicamba. So in general, as a developmental biologist, I'm really interested in figuring out what the molecular mechanisms are that are associated with auxin action. But we're also interested in figuring out kind of more broadly how auxin affects plant growth and development, especially with relationship to crops, because it is used so widely in agriculture. So auxin is produced in particular tissues via biosynthetic enzymes, and then it is actively transported either within tissues or across tissues to elicit some sort of growth response. Could be positive, could be negative. It really depends on the organ in which that it is present in. It's really important generally for plant growth. It's important for stem cell maintenance. So stem cell populations in both the root and the shoot require high auxin activity in order to maintain those cells that divide and give rise to new organs constantly, which is a hallmark feature of plant growth and development. It's also important for mediating environmental responses, response to stress, fine scale patterning of organs like vasculature within leaves, and then also mediating transitions, which are really critical for agriculture, like the switch between vegetative to reproductive phases. This sort of kind of yin and yang that I'm showing on the right here is set up by this balance of auxin being produced in certain tissues, auxin being transported, and these things lead to local maxima. And then, of course, there's signaling that happens in response to the hormone. And then downstream of that, there can be metabolic changes and mostly changes in gene expression, which is what is historically associated with auxin action. In maize, we know a little bit about different genes that are important for aspects of auxin, either synthesis, transport, or signaling. But compared to Arabidopsis, we actually know very little. So there's a lot of genetic determinants here that are missing, that we don't quite understand. Many of these different genes, whether they're important for transport, biosynthesis, or signaling, all tend to fall into fairly large gene families, conserved among land plants. So some of these things are a problem in terms of genetic redundancy. Some of these things are also just a challenge in terms of, working in a crop that requires a lot longer of a generation time for screens compared to Arabidopsis, where people have really extensively hit the auxin system pretty hard. And we know, many, many genes that are important for different aspects of auxin-mediated growth. So what I'm showing on the left here are a couple mutants that are affected in either auxin biosynthesis or transport. And you can see that it basically leads to organs which are completely devoid of lateral organs. So obviously this is really detrimental for like tassel formation and for gamete formation, where these higher order branching effects have to happen in order for successful reproduction to occur. Auxin is classically associated with organ formation. You can see that there's also some defects related to tassel branching and ear formation when two particular signaling mutants are affected, IA27, IA20. There's also defects associated with another IAA that have to do specifically with leaf growth and development. That's the Hoja loca mutant. And we actually only have one mutant that's been described to date with any auxin pathway sort of annotation for root growth and development, and that's IAA10 with rootless undetectable meristem 1 on the right. And so you can see that these roots have a very, very reduced root system architecture, no lateral roots, no seminal roots. And these things obviously would have huge effects in the field if we think about how juvenile root systems might impact crown root and later root systems throughout growth and development. So some of the kind of motivating questions here are to expand this toolbox. These are some of the well-characterized root development mutants in maize. So we want to add more genetic regulators to this list, because I think if it's really important for us to know how root system architecture can positively be obtained in order to improve nutrient acquisition, water availability, drought stress, things like that, traits that we're interested in that impact yield, then we have to know a lot more genetic components in order to flesh this out, I think, and also inform strategies. Okay, so I'm going to address these two outstanding questions today. Number one, what roles do auxin response factors, these transcription factors that mediate auxin response, play in maize root growth and development? And then the second question is, what are these other genetic regulators that we haven't discovered to date? And I have them as 1 and 2, but I'm actually going to talk about it as 2 and then 1. Okay, and what time did we start? Because I tend to ramble. 5 after. Okay, so what we wanted to do is this genome-wide association study to look at auxin responses. But in order to do these sorts of screens, you have to have a very robust, very reproducible phenotypic platform in order to be able to link genotype and phenotype associations. So this really relied on having a good rolled towel assay that we could do exogenous hormone treatments with. And this is a method that is actually in prep right now as a protocol, but was first worked out by Michelle Lang. She was an undergrad in the group. She's now at Corteva. And then Melissa and Becca have subsequently refined and expanded it. So I'm just going to mention the method briefly that we use to do our phenotyping screening and then get into the GWAS. So this is our improved rolled towel assay. Rolled towel assays have been around for a while, and they're used widely both in corn and in soy and other species as well. So we start with these seed germination papers that we buy from Anchor, label them, they're in a triplicate stack. We then sterilize our kernels with bleach. We plant them on paper, the same seed germination paper, which has been soaked in a fungicide called captain. It's a critical step. And then we roll up the towels into these little cigars or rolled towels, and then place those in a beaker, and then beakers go into some sort of controlled environment. We use a Percival, but you could also do this in a greenhouse. After 2 days and going up to 4 days, we score for germination. This is a really critical aspect of these assays because we need growth to be really synchronous. Developmental stages obviously are very, very dependent on time. And so we don't want to be imaging plants that are not at the same developmental stage because that would really skew our data. So most seed batches tend to germinate around 50% or greater within 2 days. Some can take up to 4 days. It really depends on the genotype and it depends on the quality of the seed. So we do this, and then once everything has reached at least 50% or greater germination, we call that day 1, and we remove any kernels that are not germinated so that what is growing is synchronized together, same stage. Okay, so using this sort of assay, we can do different auxin response measurements that can allow us to measure auxin phenotypes, auxin-dependent phenotypes. So here's a very simple one where we took B73 and we treated them with auxin for 2 days, starting at 3 days after germination. And you can see the auxin-treated seedlings on the right have a very, very short root because auxin inhibits primary root growth. So this is a very strong response. It's very easy to quantify this, and it's very reproducible. If we continue this sort of assay and do more of a prolonged growth, so again, starting a hormone treatment at 3 days and going up to 12 days, you can see untreated root systems have a very well-established primary root. Lateral roots are forming. The seminal roots have emerged and elongated. In contrast, when we look at our auxin-treated seedlings, you can see that the primary root is greatly inhibited. The lateral roots and the root hairs are promoted, and the seminal roots are also inhibited. And so these are the sorts of traits now that we can measure dependent on auxin given particular genotypes. We actually stop our assays much sooner than 12 days because that's a pretty long time point, but I just wanted to show this as an example of how long you can let these assays go on these paper towels. Okay, so Becca and Melissa have also done these same assays with several other hormones. So brasinolide, a brasinosteroid, cytokinin, benzylaminopurine, BAP, which is a synthetic cytokinin, and jasmonic acid. So in black is the control. These are all done in B73, the reference genome. And you can see that increasing concentrations of hormone, shown in blue, orange, or purple, for each of these hormones at different concentrations can greatly inhibit primary root elongation. So now we have in hand an ability to screen for hormone responses, specifically within the root across different hormones, and usually with one particular given concentration that seems to have the most robust, statistically significant phenotype. I'm just going to talk about auxin today. Okay, so then once we had this assay in hand, we decided to use a well-characterized diverse panel of inbreds known as the Wisconsin Diversity Panel to look at how auxin-driven phenotypes vary in both the shoot and the root. We were more kind of particularly interested in the root, but because shoots are also easy to phenotype, we also measured those. So this is a line of around 620 different genotypes, which SNPs are already available for. So it's a great panel for doing genome-wide association studies. This has been published by Candy, then Hansey, now Hirsch. So if you want to go read on this paper of what consists of these lines, the details are here. So we bulked these up in summer 2020. So Nick and Miriam and Marna Yandel Nelson and Justin and myself and Max and Dirk, it was like this whole feat for us to put out all of these lines in duplicate and bulk everything up so that we would have sufficient, really high quality material for doing screens, which were then started that subsequent winter by Becca and Melissa in the heart of the pandemic. Okay, so this is the outline of how we did these assays. So kernels were sterilized, just as I mentioned, planted by the same rolled towel assay. After 3 days, seedlings were treated with auxin. We used 10 micromolar IAA, and then images were collected when the seedlings were 7 days old. We also excised off all of the roots and froze those away at -80 to use for gene expression analyses for transcriptomic, proteomic, and phosphoproteomic studies. So it was a lot of image acquisition. It was a lot of tissue collection. My -80 is currently full of frozen roots. But we got images down below, which I'm showing a picture of. We have about 12,000 images that have been analyzed and phenotyped for both shoot and root traits. And this was actually done by manual annotation using Fiji. So you know, Becca, Melissa, Sarah, Ludvin, and John were pretty amazing to do all of that. I should say, so for each genotype, we also had at least 10 biological replicates, and then we had 10 go to a mock treatment and 10 go to an auxin treatment. So lots of informative data there. Okay, so this is the list of the traits that we quantified. So shoot length, root length. This was both done in mock and auxin treated samples. We also calculated ratios of shoot to root, and we also calculated that same ratio in response to auxin, knowing that the canonical response is that auxin inhibits root growth, and so we would expect an increase in that ratio in a normal sort of wild type situation. Okay, but we're interested in finding phenotypic outliers. So from these data, you can see that the shoot traits are very normally distributed. So we have a mean shoot length here. When we treat them with auxin, it doesn't change. So auxin doesn't seem to do much in juvenile maize shoots, at least under these conditions. And then when we take this ratio of IA to mock, we now have another representative value that can allow us to look for outliers. So you can see that there are many genotypes, which are at the top, which have a very altered auxin response. They actually do elongate quite a bit in response to auxin. And then we have some which are hypersensitive down at the bottom. So these are genotypes that are presumably deviating from the norm. And the question would be, why is that? What's sort of underlying their differences in auxin responses? Same for the root. So we have a normally distributed root length in response to auxin. This is greatly reduced. We already know that from the assays that I showed before, because auxin normally induces repression of primary root. And then again, we have this sort of centered repression, like for B73, it's around 70%. So with these values, we've dug in a little bit more to look and see what some of these genotypes are that have these abnormal responses. So as I mentioned before, for roots, I'm not showing the shoot data because I'm just going to focus on roots today. The normal response for B73 is about 0.71 for inhibition of auxin-treated roots to the mock control. We didn't see any of the NAM founder lines as showing auxin insensitivity. And on the right here, I'm showing a smattering of genotypes, which are up at the top, which all have really, really auxin insensitive responses, which is interesting to know why these things are not behaving normally. We also have some hypersensitive genotypes, only a couple of these, and we do have a slightly hypersensitive NAM founder line, OH43. Okay, so now that we have all this phenotype data, we then want to compile this with our SNP data for the WITIV population and actually do GWAS. So this is work that Raj, who's a postdoc with Brian Dilks at Purdue, has done. So he started with a lot of SNPs and ended up with a lot of SNPs, and I'm not going to go through the details of this. And then once we were down to around 20 million SNPs, we wanted to map these. So you just see a little cartoon here of how all of these SNPs might pile up along physical positions. And then we retain SNPs with an association value of at least P 10 to the -4 for these P values. We then defined a locus as a set of consecutive SNPs within a 250 kb window. And then within that 250 kb window, we would take the SNP with the lowest p-value. And then we were able to identify candidate genes by tagging SNPs that are associated with the annotated genes, either directly within the gene, or we would take 2 genes down or 2 genes up. And so this gives us our candidate gene list. This is what the data look like. So you can see here that the different traits are color coded. So mean root length is in yellow dots. Mean root length, that's in response to auxin. Mean root length mock is purple. And then the ratio of these are orange. And so we have many hits across these different chromosomes that might be of interest, especially for co-occurring traits within the same SNPs. I'm just going to mention a couple of them right now. So one of them turned out to be this auxin response factor, ARF27, which my group has already been working on. This is a transcription factor that we know to be really, really important for proper auxin responses in the root. And we actually already have mutant phenotypes that directly connect it to auxin-mediated root growth and morphogenesis. So ARF27 is required for root hair formation. In the absence of this transcription factor, root hairs do not form normally in the elongation zone. Also, when we treat these roots with auxin, we get ectopic formation of root hairs in the meristematic zone, which is a population of stem cells that normally should not differentiate. We also have quantified some other traits. This is again work from Melissa Draves and the group, looking at lateral root density, seminal root length, seminal root number, and some other traits as well, shoot length, things like that. Okay, so we also went through our candidate gene list, and then we looked to see of those candidate genes, which of those did we have available from our existing seed stocks across our groups to actually phenotype for auxin responses and root traits, because many of these sort of classical maize mutants might be available, but they haven't actually been phenotyped for root traits yet. So here's a list of genotypes that Becca and Melissa screened from the Dilks group from their collections. And you can see that there are 3, the one in pink and kind of gray blue and then mint green, which all have short roots. And then we also have a couple which have altered auxin responses. So 2 that are completely insensitive to auxin, and then 1 that's kind of hypersensitive to auxin. So these candidate genes, I think, are a very powerful way for us to validate our GWAS hits. And I think it's also a great way to assign new function to genes that are important for root growth and development that haven't previously been linked before. Okay, so next, we are continuing these hormone GWAS screens. So we have several other hormones that we're keen to screen. We also are dealing with the molecular side of the project now. And this is really in collaboration with Justin Wally, who's the proteomics guru. So we are working right now to profile not all 627, because that's just basically impossible, but a subset of around 200 of these lines, the more phenologically restricted population of the WIDIV group, for their proteome, phosphoproteome, also some other modified proteins. So keep an eye out for that. We're also going to use these data to make gene regulatory networks, and then also probably repeat this recursive process for these other hormones. I just want to mention briefly that this system is really great too as a tool for science education and outreach. So we've been doing these Science Bound Saturday events in partnership with Science Bound, and also through support of a ASPB Bloom Award that I got last year, to work with high school students, 10th graders and 11th graders, give them some experience to look at corn seedlings, phenotype seedlings, do a little bit of data analysis in RStudio, and just hopefully get them a little bit excited about the corn plants that they see probably growing in the fields around them, but don't know much about. So we've done 1 in person, 1 virtual. You can see these little kits that I put together for the last virtual event, and we have another in-person event tomorrow. I'm going to end here by saying thank you for your time. I hope I didn't go over too much. Thanks to our funding. We're supported by USDA, NIFA, NSF. I've had some other funding from the Crop Bioengineering Center, the American Association of University Women, also, of course, the College of Ag and Life Sciences. Justin's supported by the Plant Science Institute, which has been a huge part of this collaboration. And I just want to say thanks to all the people who have done all this amazing work and who have been great collaborators. And I'd be happy to take questions.
From the room, we have 1 quick question.
In the WIDIV panel, did the ones that showed hyper or hyposensitivity for any trait, was there any sort of conserved evolutionary?
Random. Yeah, we couldn't really make out any patterns of things that they had in common.
Okay, I'll go ahead and just introduce Alex briefly. So Dr. Alex Bucksch, his research focuses on the understanding of root architecture variation in the plant populations. In particular, he develops the phenotypic spectrum as a theory to categorize root architecture variation. So more roots today. He works on various crops. He uses common bean, maize, cassava as systems to develop phenotyping methods that allow for quantitative 3D assessment of the difficult to observe root architectures below ground. These methods he develops elevates the root research community at large through online platforms like Plant IT and DIRT. These platforms enable researchers without technical training to scale their research with supercomputing. He received his PhD from Delft University of Technology in the Netherlands and currently is an associate professor in plant biology, forestry and natural resources and bioinformatics at the University of Georgia. He was awarded an NSF Career Award in 2019, the NAPPN Early Career Award in 2020, and the Fred C. Davison Award in 2020. So with that, we welcome Alex.
Thanks for this nice introduction. Just here, I start off with this picture, which basically shows already the data we are working with, which is basically imaging data, either in 2D, what you see on the left side here, that is just a photo of a maize root, and on the right side is a 3D reconstruction of a maize root, which is already a bit noisy. So you can already imagine what kind of data problems we just deal with every day. So I want to start off actually why we do that. And one of the things that you know in the Midwest probably pretty well is that it's getting hotter and over time, and many of the climates you will face are already here in the southwest of the US or are already present, and you will see them. Yeah, by 2050 you will have similar problems that are now down here in the southwest prevalent. And if you want to quantify that, you see that it's mostly Missouri in this list. I just took Tucson because I have collaborators here. But what you see is these areas are already there, what we will hit in the 2040s and 50s. So there's a reason why we should think about roots and how they can improve their efficiency. And typically, if we think about how we can get more efficient roots, we think about the phenotype that is made out of the gene and environment interactions. And this phenotype, which is here on the left side, just an image in 2D, changes quite drastically with the environment that it is put in. So you have here the same genotype that is, I think it's a DOR 364, and you see that the angles of the basal roots in this common bean change a lot. You see that the upper one in the drought does not show many adventitious roots, but in the well-watered conditions, it shows adventitious roots. And these are kind of traits that if you can observe them quantitatively, we think, and we can link them somehow through genetic information, we think they are also possible to breed. So the question is, what has this now to do with data? Very obvious. So on the left side, these are already images. These are simply imaging data that we can digitally collect. We have sequencing data from the genomes, and from the environments we get normally a lot of sensor data that we can all link together to solve this G cross E problem from a phenomics perspective. What I wanted to do today, which is a bit different than we normally give our talks, is just to show you what is, from my background, when I'm training in computer science, what is the abstract data problem we are actually dealing with. And if you look at this picture here on the right, it's very noisy. It's basically a random pattern, but if you put a few very simple rules, this is here diffusion reaction model, to this initially random situation, you see simply a pattern emerge. very quickly. So if you put that further, you will see actually a whole network, could be a leaf venation network emerging. So that's one thing to think about, is simply that very simple rules can make randomness a structure, even though randomness is considered unpredictable. So if you look from another perspective, and that's here what Chris Langton basically did a lot. If you have seeds growing in the ground, for example, just some predetermined structure, but it starts interacting with each other, you can easily end up with something that looks pretty much random. So I put it a little further, and you would probably, if you analyze this final state, you would just get something that looks very random, but it has an underlying simple rule, which is just these squares that just expanded here at the beginning and started interacting with each other. Just like roots below ground, they start interacting with each other and give you kind of very chaotic data. But it's kind of predictable that it will be chaotic because of the interaction. So that brings me to the simple conclusion, because our computers, right, they are basically simple rules, which are these transistors in it, and they can generate kind of random numbers, more like pseudo-random, but comes pretty close to that, and everybody uses it every day. And what I wanted to point out is, if you have a random starting point that turns into structures, or just simple rules that start interacting with each other, that could most often lead to indistinguishable statistics. So what we analyze and how we characterize our traits might be not as easy as we normally think about. And I have one example I want to talk today from my lab. We have several of them, but one is the bean root experiment, where we basically say, hey, we always think of this like random if you look at a lot of roots in a field. So and I put just here on the right side these bean roots. There are many reasons why you should look at bean roots, but what you basically see is they look very different, and they're kind of neighbors in the field, right? And typically this is typical explanations are just, okay, there's local soil inhomogeneity, or it's simply random. So I never got, during my postdoc when I started these experiments, I never got much more answers than that. So why bean? There's one reason: it's very easy to work with in 2D. All right. So typically I got these kind of roots shown to me, and they look impressive, right? But if you just look at this, at one of these samples, that's certainly insufficient. And what we did is to say, okay, let's dig up a lot of these roots. They're kind of incomplete, right? But we get an overall idea of the shape. And we did that just over a whole grid of bean roots in this desert environment here. And we did it for 3 genotypes and in drought and non-drought conditions. What we got is this, again, a huge variation of architectures that look very differently in their shape, from going more deep to more wide. And we were simply asking the question, so what is this function of this diversity that we see there? Is there actually a function associated with it? And if we think about that question, the biggest problem we face is actually which tools do we need to develop to analyze that and observe this root architecture diversity. Simply because roots are underground, there's not much out there that we can just use off the shelf. And then sure, which mechanisms underlie this diversity? For the rest of the talk, I focus mostly on the tools, because this is where the bottleneck is at the moment. And one of the tools we developed is simply a descriptor that summarizes this root architecture in an overall shape. Very simplistically, where we say, we look at how the width of the root respective to its taproot develops over that. And that results in these curves that you see here on the right. And these curves give you kind of an overall shape idea of this root system. Now, when we do that in well-watered and water stress conditions, we actually didn't see much differences between them. So they just look very chaotic or randomly mixed through each other. And we thought, can we sort that out? Can we perhaps find groups of similar curves that lets us group these architectures that we just see in the field into more homogeneous groups that are likely the same? That's what we did. And there's an algorithm to it that was developed by my PhD student, Li-Meng Shi. And it's kind of an extension of a k-means plus clustering, where you can start clustering curves based on similarity criteria. And then there's an outlier detection to it. And because the k-means has always like a random seeds to start, we're looking at many clustering runs on a computer. And then we say, okay, what is the most likely consensus? And for the DOR 364, you see the consensus that we came up with was 5 architecture types that we could observe that look very similar, and we could group them into these 5 classes. If you look over the 3 genotypes that we had at the beginning, it looks a little different. So if you look at 5 architecture types and just say, okay, they all have 5 architecture types, we saw that this 5th architecture type is actually very sparsely present in L88 and SEQ7, which are the other 2 genotypes we looked at in the field. But only in the DOR 364, it was over the 10 or 20%. And then it's the question, like, maybe we are looking here at something different. We are looking perhaps at kind of a noise. And if you think about that, we are going over the depth and we are looking at how the width develops over the depth. You would expect normally in a 3D PCA of these slices over the depth, you would actually expect something like a spherical result. And what we came up with is that in the DOR 364, we really see this sphere on the right, on the left, and this 5th architecture type is really like a varying slice out of this sphere, or it's actually a solid sphere. And then if you look at the other 2, we see it occurs just on the outside, like as noisy sprinkles. And we concluded from that that there's simply this 5th architecture type is not present in the 2 other genotypes. So that gives us already an idea of, hey, there's a trait that might look very different. And that is the amount of architecture types that a genotype has available in its population to acclimatize to its environment. So I go one step further now and just give this small overview of idiotypes that are already known out in literature. And they're basically 3. They're forming kind of these kind of triangular or box shapes. They were published by Lynch in 2019 and 2022. They relate to different uptake functions. And these kind of shapes, they relate to the curves that we had just before. Now, that's just a current working hypothesis that we have is we see these shapes actually reoccurring if you look over these architecture types that we just had before. For example, in DOR 364, and we see there is, or we hypothesize that there is a difference in uptake within the population between the individuals. So there are idiotypes that we know, like the box or the triangle, but we also saw a lot of these diamond shapes or different kind of diamond shaped idiotypes, or even like such blobby teardrop-like shapes generally occurring that we just approximated here manually. But overall, we think that there is a transition of what these kind of root architectures are actually functionally specialized for. And we want to know now, is there any interaction that they can make use of, or any kind of these kind of questions down the line? Notably, this is all in 2D. This is a very rough look on the situation. So we are not exploiting the full architecture or the full potential of what we could. And then we ask that question, can we actually add more detail to these kind of data? So the 2D gives us a hint, more or less like an existence. There are different architectures below ground, and they relate to functions that we might know. So how do we add more detail? And that's where we come back to maize. So one thing we did in 2016 is to say, can we deal with all of these occlusions from a technology point of view that are in a maize root? And the situation was like that we used just a lot of images from all sides around the maize root, and we ended up with this kind of result. It had a lot of gaps, not really possible to analyze this structure. So what we did is we used this, it's a structure from motion technique that we resolved like this. We basically build a scanner with several cameras on it and rotate around the root system. The scanner is relatively cheap. It's just about $2,500, maybe $3,000, depending on where you build it. And it takes about 2 to 3 minutes to scan a root. And we could also, over time, now reduce the number of images we need. We just need 300 images now. And we saw, hey, there's actually a lot of resolution we can gain if we simply include a few simple rules into these reconstructions. So we use the structure from motion technique that is out there, and we put simple rules of root growth into it. Simply, a root tends to go downwards. It branches at a certain moment. So these kind of rules are in there. We also looked a little bit into neuroscience, where we got backtracking algorithms to deal with the gaps that occur within the data. And that's what we could reconstruct them. Can you actually measure in these points? Pretty much, yes. You can even measure inside this this root crown, that very dense root crown from maize. For example, the crown root angle, which is the roots that are covered by the brace roots on top, had a very high correlation. But also, if you think about the biomass or the root volume we can actually extract from such a representation, that worked out very well too, where we had a very strong correlation between the root volume, if you would start tracing every single root that we have in our data, with the biomass that we simply measure on these models on the original samples and put them onto a scale. So you see that here on the left side, where you see lines going really through every single root. There's a little difference in here. So the colored ones are parts where we started correcting with backtracking algorithms. And then you see these lines, these gray lines that really go through the actual point cloud. What we do on the right now is pretty much a similar idea that we just had before, where we said, like, we look at the width of the root over the depth. Now we look at all of these genotypes in maize and say, let's look how the root biomass actually accumulates over the depth, which is a bit of a finer grained algorithm or way to look at how a root develops over the depth. So we are now having the root hierarchy somehow incorporated in the way we describe it, because we trace every single root. We are still ignoring the scale, and we do have the density in. So we see differences between these genotypes, these 12 genotypes in this initial test panel, that are pretty strong. And we hope now in the next step to do the same kind of clustering that we did on the maize, on the bean roots, and to see, does maize also show a similar spectrum of different architectures in a population? And vice versa, can we bring the 3D to the beans and see if the 4 or 5 architecture types that we observe, do they hold up if we go to 3D, or is there actually more differences that we can group into different classes? So basically what all of this was about is we generate a lot of large data that we have to compute on. We increased a lot of phenotypic detail by putting technology onto the problem. And we started discovering these kind of novel phenotypes that are existing within the population of the crops that we study. That's not all that my lab is doing. It's just what I can do in 20 minutes. Otherwise, there is a lot of other work going on. Like, for example, one really cool project that just started is where we look at phenotypes that actually are the result of the root-root interaction below ground. There is that is just for the PhD students, things start sometimes very shaky. And this is the first prototype of a system where we could observe that. We just published on a type of a root hair that just exists during early stages of bean root development that looks like that. It's kind of hooked, kind of somewhere between root hair and trichome in early development. And we have cassava research where we can link root architecture to micronutrients in the edible storage root, and a little bit of above ground phenotyping that we just need. That is all I wanted to say for today. I hope I stood anywhere on time. And just as an acknowledgement, all the collaborators, funders that are out there, an awesome lab that helps me a lot, that is always very stimulating. And if I want you to take something home for today, then it's simply that if you want to start understanding structure and function of any kind of new phenotype that we can pull out of the data, that we might not even see immediately by eye, we definitely would need a lot of very massively scalable technology, either such a scanner and scale that one up, or any kind of new technology. And that's what I want you to take home today. Thank you. That's all I had.
Okay, let's thank Alex. Okay, any questions for Alex? Questions in the room? So we'll thank both of our speakers one more time. And thank everyone on Zoom and here today at Iowa State for joining us for Plant Phenomics Fridays.